Welcome back, fellow mitochondriacs, for another episode of Cancer as a Mitochondrial Metabolic Disease. So we, in the last episode, had talked about how we're going to be diving into the area of glycolysis, specifically aerobic glycolysis or the Warburg effect, and looking at strategies to shut that process down. We have had multiple videos on the lowering of blood glucose, or just the available substrate to the cells, so that they can't have as much available to bring in to the cells. But today we're going to be looking at the beginning of targeting each specific step along the way of glycolysis so that we can tip the scales in our favor. And I was able to find some pretty interesting information. So without further ado, let's get into it. Let me pull up my... Okay. So as we talked about in our last video, we're going to break this down systematically by kind of area along the chain of steps that is happening. And the first area to discuss are these glucose transporters, and looking at ways that we can inhibit them, downregulate them, block them, etc. And I was able to come up with quite a lot of information, giving us a good backstory of why these are important and relevant to clinical medicine, but also both natural and pharmacologic ways of potentially inhibiting them. So I'm going to use this graphic over and over again because it's a pretty decent way of looking at where we're at in this process, and very similar to the glutamine. I should just make sure I can remember what number, 924. Very similar to when we were looking at glutamine, and I kept going back over and over and again to this particular graphic so we can kind of get oriented to where we're at on the glutaminergic pathways. We're going to be doing that from glycolysis, or the breakdown of sugar, as well. So today, I've circled where we're going to be looking at today: the glucose transporters, the GLUT, and the different GLUT transporters that are potentially inhibited by a variety of substances. I kind of alluded to this the last time. A lot of these papers, looking at natural inhibitors of glycolysis, will have beautiful graphics like this. They'll have, an area of the cell. They'll have, a little blockade sign, and then it'll have, 10 different substances, kind of like here at lactate dehydrogenase A, where we have, 10 different things. And although it looks cool, it is not to the degree of specificity I'd like to have for us. So, for example, I'm going to skip to the end. We're not going to get there yet. But I was able to go through the literature and actually find, for every particular substance, high yield substance, I was able to find whether it either downregulates or blocks, or upregulates and enhances. And you may ask, well, why would we want to enhance these transporters? And because some transporters in other parts of the body, like particularly muscle cells, if we upregulate or enhance those, that can actually help us lower blood glucose while blocking other transporters that are maybe getting to the cancer. So that could also be kind of a double whammy of benefit there. So without further ado, this is a paper that was published in 2012, November, Current Opinion in Oncology, and it says glucose transporters and cancer metabolism. And it says the purpose of a review: transformed cells exhibit a high rate of glucose consumption beyond that necessary for ATP synthesis, or energy synthesis. Glucose aids in the generation of biomass and regulates cellular signaling, critical for oncogenic progression. A key rate-limiting step in glucose utilization is the transport of glucose across the plasma membrane. This review will highlight key glucose transporters, GLUTs, and their therapeutic targeting in this class of proteins. And it says GLUTs, enabling the facilitative entry of glucose into a cell, are increasingly found to be deregulated in cancer, Although cancer-specific expression patterns for GLUTs are being identified, comprehensive analyses substantiating a role for individual GLUTs are still required. Studies defining GLUTs are being rate limiting in specific tumor contexts. The identification of GLUT1 inhibitors via synthetic lethality screens, novel engagement of insulin responsive GLUT4 in myeloma, and identification of GLUT9 being a urate transporter are key advances, underscoring the need to continued investigation of this large and enigmatic class of proteins. Tumor cells exhibit elevated levels of glucose uptake, a phenomenon that has been capitalized upon for the prognostic and diagnostic imaging, aka PET scans, imaging of a wide range of cancers using radio-labeled glucose analogs. However, we have not been able to target glucose entry in a tumor cell specific manner for therapy. GLUTs have been identified as rate limiting in specific tumor contexts. The identification and targeting for tumor specific GLUTs provide a promising approach to block glucose regulated metabolism and signaling more comprehensively. So I pulled this paper first because it's the oldest that I am going to be using in this particular talk today. And I think it highlights some very important things. Number one, this transporter is critically important because it doesn't matter if you have 10x the amount of hexokinase or 10x the amount of other enzymes. If glucose cannot physically get into the cell, let's say hypothetically we're not on a ketogenic diet and glucose is abundant. If we block it getting into the cell, we're going to have some effect. And although the better strategy would likely be being on a therapeutic ketogenic diet and then looking at adding things that block this in a press pulse manner potentially, this still is clinically relevant to us. So the issue is that there's a whole host of different glucose transporters depending on the tissue, depending on the type of cancer, and it's hard to know which ones are going to be the most effective kind of across the board. And maybe in the future, maybe what they'll do is, instead of sending the cancer pathology off to the pathologist and have them looking at different clusters of differentiation, the CD markers, maybe we're going to be looking for GLUT transporters. Oh, this cancer highly expresses GLUT1 or GLUT3 or GLUT9. And then we can use specific, transport blockades in order to affect that particular cancer. I think the same could be used for glutamine. Okay, this glutamine, this cancer is dependent on glutamine. It uses highly specific SLC7A11 or SLC3A5 or whatever it is as a way to specifically target that type of cancer. That being said, we're not there yet, but I think that's where we could go to eventually. So it further says in this paper, the glucose transporter family. The GLUT's main function is to ensure that there is a constant availability of glucose for metabolism. And they do this by controlling the movement of glucose between intracellular and extracellular compartments. The genes of the GLUT, GLUT family belong to the 2A solute carrier family, and they have a gene symbol SLC2A. There are currently 14 isoforms of the GLUT genes described, and these isoforms can be classed into 3 classes based on the primary sequences. The first class, class 1, are known as the classical transporters, which are made up of GLUT1 through 4, and GLUT14, which is considered to be a duplicate gene of GLUT3. There's class 2, is made up of GLUT5, 7, and 9, and 11. And then there's class 3, which is 6, 8, 10, 12, and 13. And 13, again, is known to be the H+ myoinositol transporter. It further says, compared to the other GLUTs, GLUT1 has a high affinity for glucose and therefore plays a significant role in tissues that are highly dependent on glucose for energy. GLUT1 has been found to be overexpressed in a variety of solid and hematological malignancies. The variety of cancers in which GLUT1 has been overexpressed include, but are not limited to, large B-cell lymphoma, colorectal carcinomas, hepatocellular carcinomas, head and neck cancer, gastrointestinal stromal tumor, GIST, prostate carcinoma, thyroid carcinoma, renal cell carcinoma, lung cancer, pancreatic cancer, sarcomas, and laryngeal carcinomas. In these cancers, there are data that are for and against the concept that the degree of GLUT1 overexpression correlate with the grade of the tumor, stage of the cancer, and clinical outcome. So that's interesting. They were basically saying that although these cancers that we just read off are all highly dependent on GLUT1, they, at least at the time of this publishing this paper, which was, more than 10 years ago, they were saying that looking at pathological, if they were looking at like biopsies of these type of cancers, there was some data that was for like it showing, a worse grade of the tumor or a stage of the tumor or even like a prognosis of the tumor, whereas some data did not. I'd be curious to know if that has evolved over the last 15 years. GLUT4. So this is talking about the stimulation of GLUT4 and its subsequent uptake of glucose into tissues has been linked to the activation of the PIK3 AKT mTOR pathway. Studies in our laboratory have demonstrated a role for GLUT4 in the sustaining of multiple myeloma cell viability and proliferation. It has also been shown that in GIST cells after treatment with imatinib, there is a decrease in GLUT4 present on the plasma membrane. So this highlighting that there are different GLUT transporters present on other types of cancers. And it seems that GLUT1, at least according to this paper at this period of time, seems to be the most important of the largest variety of cancers, I would say. GLUT2 has the lowest affinity for glucose. It has been expressed in cancer lines of the small intestine, kidney, breast, insulinoma, colon, and pancreas. GLUT2 and 5 are the transporters primarily indicated in fructose transport in humans. Interesting. Albeit GLUT2 to a lesser extent than GLUT5. So they're saying that GLUT2 can both do glucose and fructose, but GLUT5 is much more responsive to fructose than GLUT2. GLUT3 is another transporter in this class, which has been relatively well studied and has been found to be expressed predominantly in the brain. So it seems like that could be very important for maybe a glioblastoma or other high-grade gliomas. It is, however, also found in tissues that have high glucose demand, such as placenta and testes. Cancer cell lines in which GLUT3 has been investigated include breast, choriocarcinoma, ovarian, colorectal, retinoblastoma, and rhabdomyosarcoma. There have been conflicting reports of the role of the tumor protein 53, P53, in the regulation of GLUT3. There has been reports that GLUT3 expression is regulated via P53 independent pathway, whereas other groups suggest that overexpression of P53, a wild type, led to GLUT3 suppression. So basically, if P53 is upregulated or enhanced, in some cancers, it led to GLUT3 suppression. Others, it had no effect. So just taking a look at the actual structure of these glucose transporters, it's pretty interesting. So they kind of have like an open hinge situation where you have glucose entering and then being input into the cell, releasing the cell. It says outward open facing configuration without glucose, outward open facing configuration with glucose, occluded state, and then inward open state after release of glucose, and then it goes back to the outward state. And then what I thought was also very fascinating, but also to some degree a little overwhelming, is the amount of various factors that have effects on the expression of these glucose transporters. So looking at it from like a very big picture, certain cytokines like interferon gamma, TGF beta, IL-1 beta, TNF alpha, basically inflammatory cytokines, can then signal through a mediator of some sort to then turn on GLUT expression. And then hormones like thyroid hormone, progesterone, pituitary hormone, testosterone, estrogen, other hormones, they also work through different chemical signaling mediators, which then affect GLUT transport. And then, of course, there's hypoxia, glucose, and other extrinsic factors that can also affect this. The next paper we're going to look at was published a couple of years later in 2016, and it's titled Glucose Transporter Regulation in Cancer, a Profile and the Loops. It says cancer cells are characterized by increased energy demand and glucose uptake. Glucose transporters, GLUTs, are regarded as one of the most important proteins controlling glycolytic flux. At the protein level, GLUTs are regulated both by expression and translocation from intracellular compartments to the plasma membrane. Many oncogenic pathways, including the PIK3, AKT, mTOR, and the HIF, or the hypoxia-inducible factor pathways, as well as mutations in p53, RAS, are involved in the regulation of GLUT function. You can see here that in this particular paper, they talk about a very interesting loop of how when the GLUTs are blocked, that leads to reactive oxygen species. Reactive oxygen species affects the local cells as well, which then leads to signal transduction and potentially leading to kind of a synergistic situation where there's like what they call the reverse Warburg effect, where lactate is, transmitted and then is brought back as pyruvate to the normal cells. This paper also had a pretty interesting graphic, and it was talking about the various trials that had gone on as early as 2016, where they were using a variety of glucose inhibitors, both drug and supplement or nutraceutical combinations with other standard of care things. For example, like this is one of the studied drug compounds that block the GLUT transporters is WZB115. We'll see it over and over again, plus cisplatin and paclitaxel, a chemotherapeutic regimen, or in some cases they were using a whole host of different chemotherapeutic regimens, plus radiotherapy, plus both natural and kind of synthetic glutamine blockers. And they were looking at, particular cancers that they were studying these things on, which I think is pretty interesting. And then again, kind of how we talked about in papers like this, there's a variety of, I guess I'd say, graphics that are cool, and they kind of like open up doors of things to look up, kind of one by one. And the issue is that these don't necessarily talk about which GLUT, quote unquote, transporter they're talking about, which may or may not be as helpful as some of the other ones, like GLUT1, GLUT2, GLUT3, versus some of the other ones that maybe are not as obviously common in some cancers. These graphics are also going to have other parts of the pathway blocked by certain. These are mostly kind of research compounds, not so much drugs, or not so much nutraceuticals, for whether it be pyruvate dehydrogenase, whether it be lactate dehydrogenase, PFK, hexokinase, glutaminase, etc. But we're going to be focusing obviously today on the GLUT transporters. So don't get too wrapped up in this. We're going to cover each one of these sequentially to look at a variety of tools we have in our tool bag for each particular step along the process. The next paper we're going to look at was published in Oncotarget in March 2017. It's titled Glucose Transporter GLUT1 Expression and Clinical Outcome in Solid Tumors, a Systemic Review and Meta-Analysis. So it says Glucose Transporter 1, GLUT1, a uniporter protein encoded by SLC2A1 gene, is a key rate-limiting factor in the transport of glucose in cancer cells and frequently expressed in a significant proportion in human cancers. And it says here later that taken together, overexpression of GLUT1 is correlated with poor survival in most solid tumors, suggesting that the expression status of GLUT1 is a vital prognostic indicator and promising therapeutic target in solid tumors. So you remember that 5 years before, where was it? That they were looking at GLUT1, and they were saying that the data was not panning out whether or not GLUT1 expression could really talk about survival, prognosis, et cetera, et cetera. But in 2017, it looks like enough data had come out to where they believe that it does actually have it's actually a very promising therapeutic target as well as prognostic indicator, vital prognostic indicator. So that as the data becomes more readily available, then we can, make better decisions. Now, for those of us who understand that cancer is primarily a mitochondrial metabolic disease, this would make a ton of sense. But for some, other people in the scientific community that believe that cancer is a genetic disease, then these findings are probably very surprising, I'll say the least. Next paper we're going to talk about is published in August of 2018, and it's titled "Glucose Transporters in Cancers from Tumor Cells to the Tumor Microenvironment." It says solute carriers of the glucose transporter GLUT family mediate the first step for cellular glucose usage. The upregulation of GLUTs have been reported in numerous cancer types as a result of perturbation of gene expression and protein relocalization or stabilization. Because they enable to sustain the energy demand required by tumor cells for various biochemical programs, they are promising targets for development of anti-cancer strategies. So you can see here that it's starting to become more clear as we go on in years in the literature that these are very important, likely targets for cancer treatment. The next paper published just a couple months after that one in 2018, titled "Targeting Key Transporters and Tumor Glycolysis as a Novel Anti-Cancer Strategy." And it says glucose transporters play an important role in cancer glycolysis to sustain cancer cell proliferation, metastases, and survival. Utilizing the knowledge of differential expression and biologic functions of GLUTs offer us the possibility of designing and delivering chemotherapeutics towards targeting tumor tissues for improved cancer selectivity. Inhibition of glucose uptake and glycolysis may effectively kill hypoxic cancer cells, facilitate drug uptake via active transportation, provides the potential opportunity to circumvent the drug resistance in chemotherapy. GLUTs as the hallmarks and biotargets of cancer metabolism enable the design and development of novel targeted theranostic agents. So I think what is being missed heavily here is that this is obviously looking at strategies and inhibiting these glucose transporters to effectively maim or potentially kill cancer cells by blocking these GLUT transporters. And I'm not sure if it's obvious to you guys, but it seems like it's at least subtly there to me that these researchers have not considered that a client or a patient, a cancer patient, would be on a ketogenic diet at all, right? Because what I can tell by some of the wording they're using is that they're concerned that there could be really downsides to blocking these glucose transporters, because a lot of these tissues tend to be highly glucose dependent, or they utilize glucose under kind of, I guess you'd call it, normal conditions. So my thought is that we could probably remove that fear to a large degree by offering up an adjunctive, but I see it as a primary strategy. That's why Dr. Seyfried calls it a press, right? Because it's a primary strategy for the metabolic management of cancer. But if we were able to get these clients and patients on a therapeutic ketogenic diet and have them be keto adapted essentially, and have the rest of the normal cells that are able to be metabolically flexible in a fat burning and a ketogenic state, then I personally believe that blocking some of these transporters would have probably little or no effects on the rest of the body. It would only affect the cells that can't have that metabolic flexibility, aka the cancer cells. The next paper, we're getting to more what do you want to call it? More relevant, I don't know, more new studies, I guess I'd say. This was published in 2022 in Frontiers of Pharmacology, and this is a fantastic paper. And I'm probably going to potentially end on this paper because I still have a ton of graphics, although maybe we can hit it all today. So this paper is titled An Appraisal of the Current Status of Inhibition of Glucose Transporters as an Emerging Anti-Neoplastic Approach, Promising Potential of a New Pan-GLUT Inhibitors. And that's kind of like an unveil a small molecule that has been developed as a drug that I don't believe is being used in oncology, unfortunately, but it potentially could be, and it could be a pretty neat addition to metabolic therapy as well. Neoplastic cells displayed altered metabolism with accelerated glycolysis. That, if you've watching this channel for even a couple of days, you probably know that's the case, right? The Warburg effect. Therefore, these cells need a mammoth supply of glucose for which they display an upregulated expression of various glucose transporters, GLUT. Thus, novel anti-neoplastic strategies focusing on inhibiting GLUT to intersect the glycolytic lifeline of cancer cells. This review focuses on the current status of various GLUT inhibition scenarios. The GLUT inhibitors belong to both natural and synthetic small inhibitory molecule category. As neoplastic cells express multiple GLUT isoforms, it is necessary to use pan-GLUT inhibitors. Nevertheless, it is also necessary that such pan-GLUT inhibitors exert their function at a low concentration so that normal healthy cells are left unharmed and minimal injury is caused to the other vital organs and systems of the body. Again, this could probably be completely mitigated by being on a ketogenic diet first and foremost, getting those other tissues that are relying on glucose in a normal diet, standard American diet, keto adapted, and then using a pan-GLUT inhibitor. But I'm going to digress. Moreover, approaches are also emerging from combining GLUT inhibitors with other chemotherapeutic agents to potentiate the anti-neoplastic action. I think that makes sense. A new pan-GLUT inhibitor named Glutor, sounds like Thor, right? Like some kind of superhero. A piperazine 1 derivative has shown a potential anti-neoplastic agent or action owing to its inhibitory action exerted at nanomolar concentrations. The review discusses the merits and limitations of the existing GLUT inhibitory approach with possible future outcomes. And it has a pretty cool graphic here. We have basically the cancer cell, we have the GLUT transporters in various locations, and then we have kind of multiple strategies, right? We have GLUT inhibitors of natural origin, and we'll talk about those in detail and kind of where they work at and what specific, transporters they block. Then there's the small molecules, some of which we will talk about, some of which are not on this sheet actually. And then there's combination approaches, and then there's additional approaches using other new technologies such as, RNAs and certain kind of like antibodies and stuff like that. But we're going to focus mostly on inhibitors and down regulators in this particular talk. So in this paper, they have a pretty amazing graphic. And I think this graphic could be potentially really helpful for us because now, unlike in 2012, 10 years before this paper was published, they didn't have the specific cancers that use certain glucose transporters down. Now they have got much more data, and they're able to say, okay, GLUT1, head and neck, brain, thyroid, pancreatic, breast, esophageal, gastric, renal, lung, cutaneous adenocarcinoma, blah, 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 blah. And it's like all the way down the list. And then GLUT2, hepatocellular carcinoma, cholangiocellular carcinoma, breast, gastric cancers. And then let's say GLUT4, breast, lung, gastric, pancreatic, GLUT5, liver, carcinoma, lung cancer, renal cell carcinoma, breast, and prostate cancer. And then all the way down to GLUT12, right? We know that GLUT13 is something kind of different, and I think it was GLUT14 is a copy of GLUT3. So that's probably why they didn't mention that. So it's just pretty fascinating that they have got it much more characterized nowadays. Again, I think what could be a cool future technology would be that basically when you, let's say you get a biopsy, they send that off to path. Path then looks at the particular GLUT isoforms and maybe glutamine transporters, glutamine, enzymes that are specific to that type of cancer that we could then make a targeted metabolic therapy to, but we're not there yet. So the next paper I'm going to show was published in December of 2023 and is titled Glucose Transporter 3, GLUT3 promotes lactylation modification by regulating lactate dehydrogenase A, LDHA, in gastric cancer. And it says here that single cell sequencing data showed that GLUT3 expression was significantly increased in primary tumor and metastases tumor tissues. In addition, GLUT3 expression was positively correlated with that of LDH expression and lactylation-related pathways. Western blotting and immunohistochemistry analysis revealed that GLUT3 was highly expressed in gastric cancer tissues and cell lines. GLUT3 knockout in gastric cell lines inhibited their metastases and invasive capacity to varying degrees. So the reason I showed this in particular is because obviously we have this, right? And I'm sure gastric cancer is likely on this particular slide somewhere. And my point is that I'm not trying to kind of rehash out what glucose transporters are on particular cancers. I think that would be too much for the level of this video, because it would just be monotonous. We'd be just reading off GLUT1 and then looking at different cancers, right? The reason I brought this particular paper to light, because I thought it was fascinating, because when these researchers knocked out or got rid of the GLUT3 transporter in this particular cancer, gastric cancer, it inhibited their metastatic potential and their ability to invade other tissues to varying degrees. So these GLUT transporters are critically important to cancer's ability to really not only sustain itself in survival, but also to invade other tissues, metastasize, which is, what generally will harm and kill patients, which obviously we don't want. The next paper, titled Modulating Glycolysis and to Improve Cancer Therapy. We're going to probably come back to this paper over and over again because it has such great information. It was published just recently in January 2023. And it says here that several studies have shown that targeting the enhanced glycolysis in cancer is a promising strategy to make them more susceptible to treatment and other conventional treatment modalities, including chemotherapy, radiation therapy, hormone therapy, immunotherapy, and photodynamic therapy. I thought that was pretty interesting that they use photodynamic therapy here as a conventional therapy. I don't consider it conventional, but I think it's pretty cool that they put that there. My point by putting this particular slide up is because it's fairly new data, right? This is January, almost February of 2023. And they're saying here that several studies, recently have shown that when we are blocking glycolysis, the breakdown of sugar, whether it be the uptake, looking at these transporters or further pathways on the line, when we're affecting those pathways, it makes cancer more susceptible to treatment with other conventional modalities. Because the reason I bring this paper up is, and we've seen this over and over again, but one of the fears that is put into patients, probably unknowingly and probably not maliciously, but there could be other intentions, is when a patient wants to do, let's say, a ketogenic diet, or they want to take some non-approved medication or supplement or nutraceutical as a way to help themselves. The cancer doctor or the oncologist will tell the patient that it's going to inhibit chemotherapy. It's going to make it not work as well, or radiotherapy, or whatever therapy they're using. And in most cases, of the nutraceuticals, of the off-label drugs, etc., that are used for cancer therapy, generally that is not true. Generally, actually, matter of fact, it's the actually opposite. It'll actually enhance chemotherapy, radiotherapy. It will enhance immunotherapy. And it will, a lot of times, even furthermore, it will unlock further, prior resistance to chemotherapy can be unlocked via utilizing some of these therapies. And I just thought this was important to put in here, because this is fairly recent data showing that These blockers of glycolysis or glucose transporter uptake channels likely only enhance the standard of care. They do not inhibit or make worse the standard of care, and maybe even allow us or my oncology colleagues to use chemotherapy in a much different way, in a much lower dose that maybe has in a more effective way. Because we'll talk about the use of sodium bicarbonate in the future in videos, and it's being studied to enhance chemotherapy because the tumor microenvironment, the lactic acid, succinic acid tumor microenvironment that's very acidic will tend to kind of chew up and eat up the chemotherapy and not allow it to get into the cell that we want it to get into, and it's going to more affect the normal cells that don't have that lactic acid shield around them. And so my point is that most of what we're talking about here is going to enhance even chemotherapy, and allow potentially in the future, if chemotherapy is even still relevant or needed, us to use much lower doses that are going to have and have more of a targeted effect on the cells that they actually are intending to kill. I'll digress. Later on in this paper, it says, the inhibition of glucose transporters, GLUT, a critical rate-limiting step in glucose metabolism, modulates the therapeutic efficacy of several drugs. GLUT expression is elevated in several types of cancers and associated with a poor prognosis, suggesting their key role in cancer cell metabolism. So again, it went from not really sure if it's going to affect prognosis or outcomes. Now you can see that they're much more definitive with their language. They say is associated with a poor prognosis, suggesting their key role in cancer metabolism. Glycolysis inhibition using inhibitors of GLUT1, when combined with routine cancer therapy or conventional therapy, has proven to be relevant in potentiating their effects in a synergistic manner in preclinical studies for several cancers. GLUT1 inhibition curbs the self-renewing capacity and tumor-initiating potential of cancer stem cells, and has a substantial significance from a therapeutic perspective. Later on, said in this particular paragraph, I cut out some of it. Combination therapy with GLUT modulators can also improve bioavailability of chemotherapeutic drugs. The co-treatment of paclitaxel with psilocybin, milk thistle, essentially a GLUT modulator, significantly improved the oral bioavailability of the drug in several in vitro and in vivo studies, and overcame a major drawback of the limited oral bioavailability of paclitaxel. A nanomedicine-based combination using GLUT1 inhibitor and chemotherapeutic agent curcumin deprived cancer cells of glucose and sensitized cancer cells to chemotherapy, induced apoptosis, programmed cell death, improved anti-tumor effects, and alleviated side effects in vitro and in vivo. Thus, combination therapy with GLUT1 inhibitors may be a rational therapeutic strategy, and could allow for low-dose treatment with chemotherapeutic drugs, providing a paradigm for high efficacy, low toxicity therapeutic options. I mean, if that isn't pretty exciting, I don't know what is, right? Like, if we're going to still use chemotherapy, if it's necessary, because we haven't knocked it out completely with metabolic therapies, if we have to use chemotherapy, this may be a whole paradigm shifting moment or movement where we can use the same drugs, but in a very highly effective and low toxic option. I mean, it sounds like an amazing strategy to me. And the paper the graphic that went along with this was looking at, again, we'll see this paper over and over again because this paper is so good and it's recent, 2023. And it's going to kind of look at mostly drugs, as you can see here. These are like research compounds. Again, I've mentioned this. You probably don't remember it, but it was WZB117. There's a whole host of different kind of compounds that are being developed in for GLUT blockade or inhibition. And then there's other obviously steps along the glycolysis and fermentation pathways. For example, the enzyme that would be here would be lactate dehydrogenase. This would be probably several steps. It could be hexokinase, probably is hexokinase looking at 2DG, but 3PO I think blocks PFK. So my point is that there's a whole host of other steps along these pathways that we will talk about sequentially, and also will have clinical relevance. similar to that of GLUT. Although for me, I think it makes the most sense to block the GLUT transporter, just because if it can't get into the cell, it doesn't matter how much of these other enzymes that are upregulated if they don't have the available substrate. So again, foundation ketogenic diet, therapeutic ketogenic diet, potentially fasting, other things. And then maybe the blockade of these GLUT transporters may be the 1-2 punch that we can do for the glucose side of things. Remember, they can also use the here's the glutaminolysis, elevated glutaminolysis, right? So elevated use of glutamine. We've had a whole other series on things that can be potentially utilized in that arena of the cancer cell aberrant pathologic metabolism. So I think that I'll probably end on this paper, and that will be a paper published slightly older, 2019. And again, one thing I want to highlight, I've been meaning to say this. Every paper that we have looked over today has not had Dr. Seyfried or his group as an author. Okay, this is like well-established science from a variety of research groups from all around the globe. As a matter of fact, I want to say the research group who did this particular graphic and this particular paper was from, I want to say China, but definitely Asia. Whereas these other groups are not at all associated with Seyfried, and they're finding pieces of the puzzle that align completely with Seyfried. So this paper was, again, published in September of 2019. Inhibition of glucose transporters and glutaminase, aka what DON blocks, what membendazole blocks, what betulinic acid, curcumin, etc. block. Berberine probably also has an effect there as well. My point is that inhibition of glucose transporters and glutaminase synergistically impairs tumor cell growth. Imagine that. We're affecting glucose and glutamine, and we're having a clinical response. We described the discovery of a very potent glucose uptake inhibitor, Glutor, which we will have a little bit of a side on probably in the next video, which targets glucose transporters 1, 2, and 3, attenuates glycolytic flux, and potentially and selectively suppresses growth of a variety of cancer cell lines. Co-administration of colon cancer cells with Glutor and glutaminase inhibitor, in this case, they're using CB839, very potently and synergistically inhibited cancer cell growth. Such a dual inhibition promises to be a particularly effective because it targets the metabolic plasticity as well as metabolic rescue mechanisms of cancer cells. So, as we talked about over and over again, and again, none of this of which I have discovered on my own. These are researchers that are out there at the bench doing great work, publishing papers, bringing us beautiful graphics so we can show these on YouTube. But my point is that this is a winning strategy. The blockade of glucose transporters and the blockade of the glutaminase machinery at some level. I do think glutaminase enzyme is very critical. I think it's probably the most important step, maybe even more so than the uptake of glutamine. Maybe. We'll see. Time will tell. But because glutamine is needing to be converted to glutamate, and glutamate is being used to make all kinds of other important, cancer-related biomass, glutathione, and the further fermentation of glutamine to succinic acid, then it just goes to show that these two are critically important. Now, you may say, well, can't we just take a Glutor? Can't we just take Glutor and DON? Would that be enough? And I might say, first of all, I don't know, because this needs to be studied to a much greater degree. But my thought is that why wouldn't you want to be on a therapeutic ketogenic diet as a first line, basis? Not only because we know that the insulin and the, IGF-1 and the other factors, glucose and protein and stuff like that, high protein and et cetera, that are kind of characteristic of the standard American diet that lead to upregulation of a lot of these pathways that will then further upregulate these GLUT transporters and the glutamine machinery. Why wouldn't we want to take that out of the equation first and then add these these particular molecules in kind of the minimal dose necessary to achieve our goal, right? The other thing is if you're not keto adapted first, then potentially the use of these, pan glucose transporter inhibitors like Glutor may be potentially dangerous. Again, clinical trials are going to have to be done in order to look at the safety of these things and also the tolerability. I'll tell you that we'll talk about Glutor in particular. Like I'll have a whole couple of slides on it and its particular, some graphics and things like that in the next video. But what I'll say is that I could not find at this period of time, March 2025, I could not find a dosing regimen or strategy to be used with it. Now, the cancer researchers and one of the papers, I can't remember which one it was saying that it's able to be used in like ultra low, what they call, I think they said nanomolar concentrations. So my gut tells me it's not going to take much. I will also tell you that you can, if you Google Glutor, you can see it's on these chem websites. Use it to, don't use anything without, talking to your doctor first or somebody who has a clue in this manner. And I would say that if anybody acts like they have a clue in this manner, I would say that you may want to like put up a yellow flag at least, because I could not find any dosing recommendations for this Glutor molecule. But I'll digress. My point is that we have a lot to learn with this. And as with any, glucose blocking or glycolysis blocking molecules, it would be highly recommended that you are in a ketogenic state and you are completely keto adapted before you start toying around with this. The reason I say that is because something that was also in this particular graphic from the prior paper was 2DG. And I will tell you that even people in therapeutic ketosis and who are keto adapted, have a good GKI, have low blood glucose, have high ketones, the whole gambit, when they take 2DG, they feel hypoglycemic. So my point is that even in a keto adapted state, you may have some effects of some of these compounds. So therefore, imagine that how bad you would probably feel if you were like eating carbs, normally, and you were not in ketosis and you're not feeling bad and you're blocking this when you're kind of like relying on that as your normal metabolism. It would probably be dangerous, I would guess. So that's why I believe that even though the strategy of the glucose and glutamine inhibitors as either drugs or as nutraceuticals or as other kind of like natural strategies are a winning strategy, I think it makes the most sense from a efficacy strategy, but also from a safety strategy to be on, again, the foundations of the Press Pulse, which is a therapeutic ketogenic diet that is going to be best for you. Now, previews of the next video. I have a bunch of different slides from various papers that have a bunch of different, inhibitors, natural inhibitors. I want to look at those first for a variety of targets, right? PFK, HK, GLUT, whatever. We're going to be looking at these very systematically. So the next video after we hit these GLUT transporters will be hexokinase, HK. That'll be the next one. And then we'll go PFK, and then we'll go to PKM2, and then we'll go to lactate dehydrogenase, and so on and so forth. But my point is that these graphics, although really cool, don't really give us the full story. It's not gonna other than this graphic, it says GLUT1 and it has a list of things, right? So my point is that I'm gonna be going over these in a very, very specific way by looking at where I could find literature and how these particular inhibitors have on very specific glucose transporters, which may or may not be relevant to your particular cancer as we understand more about what glucose transporters happen to be highly expressed on a variety of cancers, right? So my point is that, again, trying to like future think, let's say hypothetically you have, I don't know, head and neck cancer. We see that it has highly expressed GLUT1. Well, it seems like GLUT1 seems to be really the main one, but I don't know. Let's use another one. Let's use pancreatic. So pancreatic here in GLUT1. Pancreatic is in, oh, here's head and neck too for GLUT3. Sorry, I missed that. But my point is you may be able to go down and say, okay, GLUT1 and 3 are the most important for my type of cancer. So therefore, I may want to go and look at this diagram, for example, of saying, okay, GLUT1 is affected by this, this, and this, and GLUT4 is affected by this, this, and this. And my point is that's what we're going to be looking at in the next video or in the next episode of Cancers and Metabolic Disease. So let me take a look at, I've been talking now for 51 minutes. Let me take a look at if anybody's had any comments. I've been really keeping my head down here. So Alan says, watching you on the big screen today. That is in some ways scary. I hope it turns out good. Enhancing certain biochemical pathways is the key. Enhancing certain biochemical pathways. What do you mean, Descend? I'm studying upregulating specific mitochondrial peptides and anti-aging. Yeah. I think that I'm not sure if I'm necessarily on board with the peptides thing, but what I could say is that when you put yourself in a state of therapeutic ketosis and you're giving the preferred fuel to mitochondria and you're doing other things that are known to enhance mitochondrial function through the upregulation of PGC1 alpha and all these other, effectors that lead to mitochondrial biogenesis and or mitophagy in the normal cells and the normal tissues, then I would say that, yes, you're going to improve your situation. Agreed. And you're going to probably allow those other cells to, suck in required nutrients that potentially could starve the cancer cells. That I will say. Dr. P, longtime follower of Seafried, and now you. I have a friend with 20-year-old daughter with glioblastoma. I'm so sorry to hear that's horrible. You are also helping me with how to avoid angiogenesis-related VEGF and retinopathy. I'm glad. Hoping you could discuss on how to recover mitochondrial health. I was once a national class cyclist, but now sometimes I can barely cross the street. I know it's mitochondrial dysfunction. So I have a whole little micro series on mitochondria. In particular, you may find it useful to look at the videos on mitochondrial biogenesis and mitophagy, mitochondrial specific autophagy, and then possibly also another video that could be helpful for you is there's a whole micro series on mitochondrial heteroplasmy. And what we'd want to do is we'd want to reverse your mitochondrial heteroplasmy to the greatest degree possible so that we could, hopefully get you back some stamina and function. And maybe you wouldn't be national class cyclist, but maybe you can get you back on the bike at least, and I suspect you miss that and enjoy that. And that's also part of your psyche too. And your quality of life too. Hi, you are helping me so much. Thank you, Dr. Peebler from Texas. Well, you're welcome from Florida. Thank you. I'll look indeed. Okay, good deal. All right. Well, if there's no other further questions or comments, I'm going to probably take us offline here then. If you like videos like this, please like it for our algorithm purposes. Share us a comment, even if it's just a thank you or whatever you're thinking about these particular videos. It really helps. If you're not a subscriber yet, please do subscribe. We're happy to have you as part of our mitochondriac army. And then lastly, if you know of anybody, like particular friend here, Jeff, who said that he has a 20-year-old daughter, friend who has a 20-year-old daughter who has GBM, and you believe that there's somebody who could be helped by these videos, please feel free to share it with them. And a couple of last-minute comments came in. Thanks, Dr. Peebler, your work and research and support with metabolic therapy, and Dr. Seafried. Thank you. You're welcome. I have a lot to learn, everyone. I don't pretend to know it all. Every time I do a video and I dig into the research, I learn all kinds of new stuff, and I know that I'm going to also get a benefit when I start doing podcasts with high profile individuals. I really want to I haven't asked him yet, and knowing that he's on podcasts all the time, I hope he'll say yes, but I suspect he will. Dr. Seafried, I'd like to have him on the type. Thomas Duraj. Tomas actually told me he would in December. So once I get these podcasts up and running, I'm going to hopefully learn from the real experts in this field. Dr. Dom D'Agostino, there's a whole bunch of people that I'd like to interview and get their particular experiences, whether it be in the lab or whether it be, at the bedside and with patients and their clinical experience, of which I would love to, absorb all that information, energy, and experience and have it enhance these videos as well. I appreciate it, Jeff. He said that you're really good at explaining down to us. It's all very usable. That's interesting you say that, because I think a lot of people, you know, if they were to come into a video like this at the very beginning, they're going to be, I mean, I get comments, trust me, I know how it goes. They're going to get fairly turned off because we do go into the weeds in this channel. And I'm not ashamed by going into the weeds. I actually think that in these type of a scenario, you know, in integrative medicine in general, it's probably a good idea to get into the weeds and get into the studies, because you're already going against the machine and the grains. But especially within, you know, integrative oncology, or, you know, metabolic oncology, you may call it, basically against the standard of care, or not against, but you know, not just toting the company line of the standard of care. I think it's important to really look at the inner workings of what we're trying to explain here in a very scientific manner, so that when your doctor or your oncologist sees what you're doing or what you're wanting to do, and they look at what we're presenting here, if they're willing to, it's really hard to argue with, like, I mean, how many papers have we published, you know, looked at published papers at this point in the series? You know, we're a year into this series. I would have to say we probably looked at thousands of papers at this point. Like, thousands of papers are leading to this being the truth, right? So it'd be different if it was a one-off, you know, paper, you know, okay, like whatever, one paper in a rat or whatever. But these are like hundreds and thousands of papers that we're looking at that really help us here. I appreciate you saying that about the larger audience. I'm sure it'll come with time. Dom will be a good guest. Energy pathways are a specialty for him. Of course, that's why I want to have him on. He's like the ketone expert, you know? And I would love to pick his brain about different ketones, potentially different types of ketone esters, or different ketones that could potentially be more effective than just what you get, you know, by buying beta hydroxybutyrate or other things. Obviously, he's also an expert in hyperbaric oxygen as well. I'd like to pick his brain on that as well. So that's one thing I want to do, is I want to have, my goal is, when I do these podcasts in the probably the near future, is I want to have a lot of questions that I want answered, and other people who have asked me questions over and over again that I don't have the answer to, I don't know the answer to, that I'd like to at least ask them to see if they have an answer that would be helpful. Standard of care is a misnomer. It should be called a popular guess. I agree. Look, that's, science is always a popular guess, right? But the problem is, when industry gets involved and they start to demonize other things, and they start to like prop up theirs, and kind of a disinformation campaigns, and all the money and power and influence goes into one area of science, that's not really science anymore. All right. Well, we've gone 58 minutes. I think it's long enough. Again, if you like these videos, please share it with other people that you potentially think that they standard of don't care. I can understand why someone would say that. All right. Until next time, everybody, God bless.